Welcome to Rad Onc Smart Review, the RadBio edition.
Great to be back. Yeah, ready to get into the nitty-gritty molecular details that are just so important for boards and honestly for everyday practice too.
Absolutely. Today, we're zoning in on two really pivotal molecular pathways. You could call them the cell cycle's gatekeepers.
We're talking p53, p21, and E2F. These guys are essential. They make sure cells don't just, divide when their DNA is messed up.
Which, as you know, is a massive deal in radiation biology.
Yeah.
Damaged DNA needs sorting out before replication.
Exactly. So we'll walk through how p53 gets woken up by DNA damage, how its activity is kept in check, and how it uses p21 to basically hit the brakes on the cell cycle.
And then we'll unpack the E2F pathway too. It's another key controller specifically for that G1 to S phase transition. We'll look at how CDKs regulate that whole process.
This is really fundamental stuff, foundational, especially if you're prepping for boards right now.
Okay, let's get into it. So let's start with the star player, the guardian of the genome, p53.
Right, a well-deserved title. Now, in a normal, unstressed cell, its levels are actually kept quite low.
Yeah, you don't see much of it around. Why?
Because it's constantly being broken down, degraded, but then you introduce DNA damage like a double-strand break from radiation.
And bam, p53 levels shoot up fast.
Very rapidly.
So how does the cell pull off that quick stabilization? It's not just making more protein from scratch, right?
No, not initially anyway. It's mainly about stopping the degradation, and that happens through post-translational modifications. Phosphorylation is the big one here.
Okay, so something detects the damage first.
Exactly. You've got damage sensors. Think of the MRN complex. That's MR11, Rad50, NBS1 for double-strand breaks. It senses the break and then calls in the cavalry.
The kinases.
The kinases. Like ATM, ataxia-telangiectasia, mutated. ATR-ATM and Rad3 related is also involved, especially for other types of damage or replication stress. And DNA PK, the DNA-dependent protein kinase, plays a role too.
And these kinases, especially ATM for those double-strand breaks, they directly target p53.
They do. They phosphorylate p53, adding phosphate groups onto specific amino acids like serine 15 in human p53.
And that phosphorylation, what does it actually do?
This is crucial. It disrupts the interaction between p53 and its main negative regulator.
MDM2.
Exactly, MDM2 or HDM2 in humans. This phosphorylation physically blocks MDM2 from binding effectively to p53.
And if MDM2 can't bind?
Then p53 isn't tagged for degradation anymore. It stabilizes, it accumulates in the nucleus. It's suddenly present in high amounts.
Got it. So blocking the blocker.
Pretty much. And it's not just stabilization. These modifications like phosphorylation and also acetylation by coactivators like P300CBP.
CBP being CRE binding protein.
Right. These also boost p53's ability to actually bind to DNA and switch on its target gene, so it's stabilized and activated.
Okay, so let's trace that sequence. DNA damage. That leads to APM or ATR activation.
Yep.
Which phosphorylates p53.
Disrupting MDM2 binding.
Leading to p53 stabilization and accumulation.
And then p53 gets to work as an active transcription factor. Okay, let's zoom in a bit more on MDM2. It's the principal antagonist, right? The main off switch for p53.
Right, so it binds directly to p53, specifically that N-terminal domain we mentioned.
Yep, and that binding does two key things. One, it physically masks the part of p53 that activates transcription. So p53 can't turn on genes when MDM2 is bound.
Okay, it silences it.
And two, maybe even more importantly for levels, MDM2 is an E3 ubiquitin ligase.
The tagging enzyme.
Exactly. It slaps ubiquitin tags onto p53.
And ubiquitin is the molecular kiss of death, usually.
Pretty much signals, take me to the proteasome for destruction. That's how p53 levels are kept so low in cells that aren't stressed. Continuous tagging and degradation.
But wait, there's also that feedback loop you mentioned earlier.
Yes, the plot thickens. The MDM2 gene itself, its transcription is actually turned on by p53.
So activated p53 makes more of the protein that degrades it. That sounds weird.
It seems counterintuitive at first, but it's actually quite elegant. It's a negative feedback loop.
How does that help?
Well, think about it. Once the DNA damage is repaired, the signal from 8-MAT starts to fade. The phosphorylation on p53 decreases.
Okay.
But now, thanks to p53 having been active, there's extra MDM2 protein hanging around.
So as soon as p53 loses its protective phosphorylation,
the waiting MDM2 can bind efficiently again and start degrading p53, helping to turn the whole response off quickly. It allows the system to reset.
So the disruption of that p53 MDM2 binding by phosphorylation is absolutely critical for getting the damage response going in the first place.
Couldn't happen without it. And it's worth mentioning another protein here too, ARF. p14 ARF in humans.
Right, from the INK4A ARF locus. What does ARF do?
ARF can also activate p53, but usually in response to different signals like oncogenic stress, think hyperactive growth signals.
And how does it activate p53? Does it phosphorylate it?
No, it works differently. ARF binds directly to MDM2.
Okay.
And by binding MDM2, it basically sequesters it, pulls it away from p53, preventing it from degrading p53.
So another way to disable the MDM2 break, but triggered by different stresses.
Exactly. It's another layer of protection.
All right, so we have activated stable p53 in the nucleus. It binds to specific DNA sequences, these p53 response elements.
Yep, in the promoter regions of its target genes.
And turns those genes on. Now, a really key target gene for cell cycle control is CDKN1A.
That's the one. p53 strongly cranks up the expression of CDKN1A.
And CDKN1A encodes the protein p21.
Also known as WAF1 or CIP1. You'll see all those names used.
P21 is famous. It's a potent cyclin-dependent kinase inhibitor, a CKI.
Right. And you remember, CDKs are the engines driving the cell cycle. They need their cyclin partners to work.
So in G1 phase, which CDKs are running the show?
You've got cyclin D paired with CDK4 and CDK6 early on. And then later, as you approach S phase, it's cyclin E paired with CDK2.
Okay, cyclin D CDK46 and cyclin E CDK2, the G1 and G1S engines.
And p21. It binds to and inhibits the kinase activity of all those complexes.
So just slams the brakes on them.
Puts the molecular handcuffs on.
Yes. And by inhibiting these specific G1 and G1S CDKs, the result for the cell cycle is
G1 arrest. The cell stops right there. It can't proceed into S phase because the engines needed to push it past that checkpoint or shut down.
This is the p53-mediated G1 checkpoint we talked about.
Precisely.
And the purpose, in the context of, say, radiation damage, is crucial. Why stop there?
To buy time. Time for the cell to repair that DNA damage before it gets replicated in S phase.
Because replicating damaged DNA is bad news?
Really bad news. Leads to mutations, chromosomal instability. Basically, the stuff cancer is made of. So the G1 arrest gives the DNA repair machinery a chance to work.
And if the repair is successful,
then the p53 signal might decrease, p21 levels drop, the CDKs reactivate, and the cell cycle can resume.
But if the damage is too extensive, too much to repair,
then p53 can push the cell towards a different fate, often apoptosis, programmed cell death. p21's main job here is the temporary stop sign, the arrest.
Does p21 do anything else?
It does have other roles. It can directly inhibit DNA replication by binding to pCNA.
Proliferating cell nuclear antigen, the sliding clamp.
Right. And it also contributes to maintaining arrest in G2 phase sometimes. But its fame really comes from being that key p53 inducible inhibitor of G1 CDKs. Okay, let's pivot now to the second major pathway guarding that G1 gate. That's the retinoblastoma protein, RB, and its partner, E2F.
RB and E2F. So E2F transcription factors, what do they control?
They're the master regulators of S-phase genes. They turn on everything you need for DNA replication. Think DNA polymerases, enzymes for making nucleotides, and even key cyclins like cyclin E and cyclin A.
So you absolutely need E2F active to enter S phase.
Definitely, but in early G1, E2F is kept on a tight leash by RB.
How does that work?
In early G1, RB is mostly hypophosphorylated, meaning it has few or no phosphate groups attached. In this state, RB binds very tightly to E2F factors, typically E2F1, 2, or 3, usually complexed with DP partners.
So RB basically sits on E2F and holds it captive.
Pretty much. But it does more than just sequester it. The RB-E2F complex actively represses those S-phase genes. It recruits factors like histone deacetylases, HDACs, to the gene promoters. HDACs remove acetyl groups from histones, causing the chromatin to condense up, making it hard for the transcription machinery to access the genes. So S-phase genes are actively silent.
Okay, so hypophosphorylated RB is a repressor. What tells it to let go?
Growth signals. As the cell receives signals telling it to divide, it progresses through G1, and those G1 CDKs we mentioned get activated cyclin D CDK46 first, then cyclin E CDK2.
And these CDKs, they target RB.
They do. They phosphorylate RB progressively, adding multiple phosphate groups. This is often called hyperphosphorylation.
So phosphorylation is the switch again, just like with p53-MDM2, but here it's inactivating the repressor, RB.
Exactly. When RB becomes hyperphosphorylated, it undergoes a shape change, and that change causes it to release E2F.
And free E2F is now active.
Yes. It can bind to the S-phase gene promoters and turn them on, producing all the proteins needed for DNA synthesis.
And this release of E2F, that's a critical moment, right?
Yeah, absolutely. That's the commitment point. It's known as a restriction point, or R point. Once RB is hyperphosphorylated and E2F is released, the cell is typically committed to entering S phase and completing the cell cycle, even if growth signals disappear. There's no turning back easily.
And this pathway is also controlled by CKIs, the inhibitors.
Oh, definitely. Think about p16 on K4A. Remember, that comes from the same locus as ARF.
Right. What does p16 do?
p16 specifically inhibits CDK4 and CDK6.
So by inhibiting CDK46,
It prevents the initial phosphorylation of RB by cyclin D complexes. So p16 helps keep RB in its active hypophosphorylated E2F binding state. It reinforces the G1 break.
which explains why losing p16 function is so common in cancer. It takes the brakes off RB phosphorylation.
Precisely. And p21, the p53 target, also plays a role here.
How does p21 fit into the RB pathway?
Well, p21 inhibits cyclin E-CDK2, which is responsible for the later stages of RB phosphorylation. So when p53 activates p21 in response to DNA damage, p21 not only stops general G1 progression, but also helps keep RB hypophosphorylated, clamping down on E2F activity.
So p53 through p21 puts the brakes on both general CDK activity and specifically helps keep the RBE2F brake engaged.
Exactly. These pathways are tightly interwoven, both converging on controlling that critical G1 to S transition. RBE2F is more about responding to growth signals and passing the R point, while p53-p21 is the emergency brake for stresses like DNA damage.
Okay, this is fascinating molecular machinery.
Yeah.
But let's talk about why it's so incredibly relevant clinically, especially in radiation oncology.
Right, because when these pathways go wrong, that's often the root of cancer, uncontrolled cell division.
Let's start with p53. You said it's mutated often?
Incredibly often. The TP53 gene is mutated in something like over 50% of all human cancers. It's the most commonly mutated gene.
Wow. And what's the consequence for a cancer cell if it loses functional p53?
Huge consequences. First, it loses that critical G1 checkpoint. So if its DNA gets damaged, say by radiation or chemo, it doesn't pause to repair. It just plows ahead into S phase, replicating damaged DNA.
Leading to more mutations and instability.
Exactly. And second, it loses a major trigger for apoptosis. p53 is crucial for telling severely damaged cells to self-destruct. Without it, cells are much more resistant to dying, especially in response to DNA-damaging treatment.
So tumors with mutant p53, they often have a worse prognosis.
Generally, yes. And they can be less responsive to traditional radiation and chemotherapy that rely on inducing DNA damage and apoptosis.
So knowing the p53 status of a tumor could potentially guide treatment decisions.
Absolutely. It gives you important clues about the tumor's likely behavior and sensitivity, and it's why there's so much research into finding ways to target mutant p53, or in tumors where p53 is normal but suppressed, like by high MDM2, trying to reactivate it, maybe using MDM2 inhibitors.
Makes sense. What about the RB pathway? Is that disrupted often too?
Just as often, if not more universally, when you consider all the ways it can be broken.
How so?
Well, you can have direct mutations in the RB1 gene itself that's classic in the retinoblastoma, osteosarcoma, small cell lung cancer. But much more commonly, the pathway is disrupted upstream of RB. Things like overexpression of cyclin D, or amplification of the CDK4 gene, making those kinases hyperactive, or, very commonly, loss of the inhibitors, especially silencing or deletion of the p69K4A gene.
And no matter how the pathway is broken, the end result is the same.
Pretty much. Uncontrolled RB phosphorylation. RB can't bind E2F. E2F is constitutively active. S-phase genes are always on. The cell loses that restriction point control and just keeps dividing.
And understanding this has led to actual drugs.
Yes, absolutely. The development of the CDK4/6 inhibitors, palbociclib, ribociclib, abemaciclib, is a direct result of understanding this pathway.
And how do they work again?
They specifically block the kinase activity of CDK4 and CDK6.
So they prevent RB phosphorylation by those early G1 CDKs.
Exactly, which keeps RB hypophosphorylated, bound to E2F, and forces the cell into a G1 arrest.
And these have been successful, particularly in some types of breast cancer.
Hugely successful in ER-positive, HER2-negative breast cancer. And here's a clinical pearl. Their success highlights that these cancers, often driven by cyclin D signaling, become addicted to phosphorylating RB. They need to overcome that RB break to divide, so the inhibitors don't just slow them down, they exploit this dependency.
That's a fantastic insight. So the tumor's own alterations create a vulnerability.
Precisely. Which also means that if a tumor lacks functional RB altogether, these drugs won't work because there's no RB break for them to restore. So again, pathway status matters.
It really drives home how knowing the molecular details, p53 status, RB pathway integrity isn't just academic. It impacts prognosis, treatment selection, understanding resistance.
It's fundamental. These G1 gatekeepers are critical barriers preventing cancer. The fact that they are so frequently broken in tumors tells you how important they are and why they're such key therapeutic targets.
All right, fantastic overview. Let's shift gears slightly into a quick board review blitz. Solidify these concepts for exam day.
Perfect. Let's test that recall. Ready?
Bring it on. Question 1. Stabilization and activation of p53 following DNA damage is primarily achieved through A. Increased transcription of the TP53 gene. B. Decreased ubiquitination mediated by MDM2, which is inhibited by p53 phosphorylation. C. Direct bonding of DNA fragments to p53. Or D. Cleavage of p53 by caspases.
Okay, thinking this through. A. Increased transcriptions happens, but it's slower. The rapid response is post-translational. C. Direct DNA binding is p53's function, not its stabilization mechanism. D. Caspase cleavage is related to apoptosis downstream, so it has to be about stopping degradation. That's MDM2, and MDM2 is stopped by phosphorylation of p53.
Exactly. So the best answer is B. Decreased ubiquitination because phosphorylation blocks MDM2 binding. Nailed it.
Okay, next one. The protein p21, aka WAF1, aka CIP1, a key transcriptional target of p53, causes G1 cell cycle arrest by A. Directly phosphorylating RB, B. Activating DNA repair enzymes, C. Inhibiting the kinase activity of cyclin-E-CDK2 and cyclin-D-CDK4-6 complexes, or D. Sequestering E2F transcription factors in the cytochrome.
All right, p21. What is it? It's a CKI. Cyclin-dependent kinase inhibitor. So its main job in arrest is inhibiting CDKs. A, phosphorylating RB. No, that's what CDKs do, and p21 stops them. B, activating repair. Indirect effect, perhaps, but not its direct arrest mechanism. D, sequestering E2F. That's RB's job.
So it has to be.
C, inhibiting the G1 and G1S CDKs. That's its core function for G1 arrest.
Correct. Question 3. In the RB-E2F pathway, progression from G1 to S phase is facilitated by A. Hyperphosphorylation of E2F by CDK2, B. Binding of P16I and K4A to RBC, phosphorylation of RB by cyclin-D-CDK46 and cyclin-E-CDK2 leading to E2F release, or D. Degradation of E2F by the proteasome.
Okay, G1 to S progression via RB-E2F would need E2F to be free. A, hypophosphorylation of E2F is in the mechanism. B, P16 binding prevents progression by keeping RB active. D, E2F degradation would stop progression. So it must be about inactivating RB.
And how is RB inactivated?
By phosphorylation, by those G1 CDKs, which causes it to release E2F. So answer C.
Excellent. Last question for the Blitz. MDM2 is a critical negative regulator of p53. How does p53, in turn, influence MDM2? A. p53 directly phosphorylates and inactivates MDM2. B. p53 sequesters MDM2 in the cytoplasm. C. p53 transcriptionally upregulates MDM2 expression. Or D. p53 directly cleaves MDM2 via caspase activation.
Right, that negative feedback loop again. p53 doesn't directly phosphorylate or cleave MDM2. It doesn't sequester it in the cytoplasm as its main regulatory interaction.
The loop works at the level of gene expression.
Exactly. p53 acts as a transcription factor for the MDM2 gene itself. So no answer is C.
Perfect. See how breaking down the question, identifying the core players and their direct actions helps navigate these choices. That's key for the boards.
Okay, so to quickly recap this crucial material for your RadBio boards.
We focused on those critical gatekeepers of the G1 to S phase transition, p53, p21, and RB-E2F.
We saw how DNA damage activates and stabilizes p53, mostly through phosphorylation by ATM or ATR, which crucially stops MDM2 from degrading it.
Right. And then activated p53 turns up the production of p21 WAF1 CIP1, which is that potent CKI.
And p21 enforces G1 arrest by blocking the key G1S CDKs, cyclin-D-CDK46 and cyclin-E-CDK2.
In parallel, the RB-E2F pathway controls the restriction point. Hypophosphorylated RB binds and silences E2F, preventing S-phase entry early in G1.
Until growth signals activate the G1 CDKs, which hyperphosphorylate RB, causing it to release E2F.
And free E2F turns on S-phase genes, committing the cell to divide.
And remember, disruptions in either of these pathways are central to cancer development and fundamentally influence how tumors respond to treatments like radiation and targeted drugs.
Mastering these molecular controls is absolutely key for your understanding and for board success. Keep reviewing these pathways, draw them out, talk them through. They are truly fundamental. And listen, preparing for these exams is tough. It's a marathon, but breaking down complex topics like this step by step, you can definitely do it. You have the ability to master this material.
For more practice, including complete practice oral boards, check out radoncSmartLearn.com.
Keep up the great work. Keep connecting these pathways to what you see in the clinic. And remember how central cell cycle control is to everything we do in radiation oncology.
Thanks for tuning in.